Good evening, all. So I hope you all can hear me. Am I audible to you?
Yes, sir.
Okay. So,
Yes, sir.
okay. Hi, Jonty. Can you hear me right now?
Yes, sir, I can hear you.
Okay. So, initially, I will show you what software you have to download. So, just go to all these things, install it. Okay. Meanwhile, I just wait for 2 to 3 more minutes so that rest of people can also join. Okay. So, the very first software that you have to install is it's MGL Tools. So actually this is a package of all the docking software that we required for the analysis part. So you have to install this thing MGL tools. I just search here MGL tools and just click the first one. And meanwhile just search on the second one till it is second software that we require is Discovery Studio. And open back, and the fourth software that we require is Strawberry Pearl. Yeah, this one. So these four software that we require as of now, and using these four software we can perform the docking. Okay, the initial docking that we require. So, for the Strawberry Pearl you have to click here Strawberry Pearl for Windows, and you have to download for Windows. It's free. And try to download all software in your Windows C drive. Okay, because docking require a larger space, so you have to install everything in your Windows C. And make sure your MGL tools, your OpenBabel and Strawberry Pearl must be follow the same path, the Windows C path. So here you can click on Strawberry Pearl, this 64-bit one, and it directly install, and now you have to execute that function. So I have already installed, so I'm not going to open Babel. You have to click on the first one, Open Babel download. Okay, so this is the MGL tools one. So can you see here downloads option? Just click here download. So this is for the Discovery Studio. Just click on the first link to install the Discovery Studio. Okay, and this is for OpenBabel. You have to click on download and please try to save it in Windows C. And then you have to execute that file which you have installed. So I hope you got it, what all these four software that you have to install. And just let me know, did you understood? You understood how to install all these four software?
Yes.
Okay, so now I'm going from the basic initial part that we require. So as you know, for docking, we require two. Either we can go for ligand and protein, either we can go for protein, either we can go for DNA or DNA transcript factor and like that. So it means you require the two things that you want to dock. Dock means just simple binding. If you don't understand, docking means just a simple binding. But how this binding will take place? So we have to computationally predict. We have to know that which binding is strong. For example, if I'm giving you two object A and object B, and I just say dock these two compound, and you don't know whether these compound how they will dock and each and everything. So you have to predict certain sites on compound A. Okay, that what are the active sites there that on which any other compound can bind. So that is what similar to active site prediction of your protein. Similarly, you have to also transform your two compound into different form, because maybe it may be happen that compound that you got in that existing form, they are incompatible for binding. Therefore, you have to convert into certain form that requires for the binding part. Therefore, we convert our ligand and protein into PDBQT form, because all the MGL tools, Strawberry Pearl, they reads only the PDBQT files. They are not compatible with PDB files for docking. Even the Perl script that we will use, they also require the PDBQT file. Okay, and PDBQT file contain more informations than PDB file. You can open your PDB file and PDBQT file in Notepad, and you will compare the informations. Okay, that what is the difference between these two files. Then you will get to know that PDBQT file contain the torsion angles. They consist of one more domain, which is of angle, bond angle. Each and every information is there. File is not PDB file is not like that. Okay, there is limited information. So once you have converted your molecule, now they are ready for the docking part. You have to draw some, grid box over that region. Docking will be performed. So these are the steps that we have done so far. And from Monday onwards, we will try to complete the first docking. Like on Monday, we will done the first docking, and then we will go in technical things in detail, that how we can improve the scores and what different way of docking of 1,000 ligands or infinite number of ligands with a single protein, or just vice versa like that. Okay, so these require quite more study and understanding, so therefore you have to understand some basic parts. So for docking, as I mentioned, you require two things. One is your protein and second is your ligand. For protein, you have to go to the PDB, which is Protein Data Bank. This is a database which contains protein that ever you have known so far. Every protein which has been reported so far all over the world is mentioned here. So you can directly search your protein name and download the PDB file of your protein. Either you can also go for the UniProt. UniProt is also like the same, but that also consists some gene things and some more information. And this is for the protein. And for the ligand, you have to go UniProt. Sorry, for ligand you have to go PubChem, not UniProt. UniProt is for protein. So PubChem is a database which consists all your ligands, compound, phytocompound, every information is there. So from this database, you will get the informations of ligand that you require for the docking. And from the PDB you will get the your protein that you require for the docking. So come back to the PDB, just see whether the search has been come or not. Yeah, so you can see this is the interface of the PDB, and you have to search here which protein molecule you want. If you know a specific any protein, you can type your protein name here, or you can also go for literature survey and find any disease that you have studying, or that you have been studied so far, like dengue disease, chikungunya, coronavirus, like many diseases has been reported so far. So you can discover a compound, a novel phytocompound, or you can novel drug against that disease. So for that you have to know the pathogenic proteins. You have to find out the genes or proteins that cause that disease. Okay, so that protein you have to search here. So you have to download the PDB file of that protein, prepare its structure, and then go for the docking. So we have taken a protein, RNA-dependent RNA polymerase. So this is a viral protein that causing the disease especially in coronavirus disease like COVID-19. You have been understood this thing as well. And remdesivir is a drug which has been reported for this, you know, reported for this thing. So I just simply type RNA dependent RNA polymerase. So now it will give me a certain results, and from all those results I have to select the appropriate protein. But how I can select the appropriate protein? That the structure, look at the structures, the method through which the structure is predicted, the angstrom values, you know, all these things we have to study about it. Or if you have working on any novel protein, if you are a PhD student or master student, mostly PhD student work, they work on a certain novel protein which sequence is not on PDB. Then you have to predict your sequence using the you know, structure prediction tools like Swiss model, AlphaFold, like that, and then download the PDB's form and then go for a Ramachandran plot validation. After that, you can take that protein for the docking part, but that case you will only get once you are in a PhD or you know, doing certain extraordinary things like that. So I don't know why the speed of. Okay, once you download your PDB and file of your protein and STF file of your drug should be downloaded in STF form. Remember the word STF file, okay? And your protein should be in PDB file. After that, you require some analysis of your protein. So for that, we require the Discovery Studio. So this take quite longer time to open this Discovery Studio. And once it open, we have to open our protein file there. So this is the interface of the Discovery Studio. So first of all, we have to open our protein file that we are going to download from PDB and see how many chains are there, where are the binding pockets, each and everything we will try to explore from the Discovery Studio. And once your docking has been done, again you have to come back to Discovery Studio where you have to visualize your result. Like visualize the interaction, how your drug is interacted with your protein. You can find out the residues on which your drug is interacted. You will get the diagram and that diagram will go for the research paper publication. Okay. So that thing you can analyze using the Discovery Studio or PyMol. So for opening any file, you have to click on file option, then go for open. This is the first way. Either you can directly drag your file. If you have your any file, you can simply drag it here. Okay. So this shows I have already many proteins file, but I will try to show you. I have already make a Bioticon folder earlier. Inside I have downloaded the same protein, RNA dependent RNA polymerase. So yeah, meanwhile you have to do one more thing. You have to create a Bioticon folder into your Windows C drive. Inside Bioticon folder, you have to create two more folder. One is ligand, second is protein. Inside the ligand, you have to place your drug file, which is STF file that you downloaded from the PubChem. And inside the protein folder, you have to place the protein, which you have downloaded from the PDB. And these are the other two things that I will let you know later. Come to the protein folder, open this protein, and this is my protein. I will try to open this protein and see the internal structures of these proteins and each and every information that I will want to explore about this protein. So I will just open this protein in Discovery Studio. This is the same protein which I have downloaded from the RNA dependent, like from PDB. So this is the structure of my protein. Can you see some red droplets is there? So these are some water molecules that cause, hindrance of binding of the drug. So that we will delete while preparing this structure using the MGL tools or Autodoc Vena, or also once we convert into PDBQT, these water molecules will be removed. Now what we have to do, just click on this button. This is an arrow button. Just click here. You will see some more information like this. So this shows that this protein is consist of only one chain, which is A. If you can see here multiple alphabets letter like A, B, C, D, E, F, G, H. So that means your protein is consisting of different kind of chain like A chain, B chain, C chain, D chain. Ideally, I am repeating the again, ideally you require one chain for docking. Okay, if you are taking multiple chains, you will get different score for multiple times if you're going to perform. And the best thing is that if you submit your result for research paper publication, the reviewer will perform your docking, same docking which you have performed, with the same grade file dimension. I hope you got it what is grade file if you have not attended the previous class, but just wait for it. They will use the same configuration whatever you have done, and if you are they getting different result, your paper will not be published. Therefore try to use a single chain because that gives you a fixed score, and also it has a greater impact of the publishing and all that. Okay, so here you can see your protein is consist of chain A, some heteroatoms, some water molecules, protein groups, and ligand groups. So we don't want ligand group. Ligand group is what? Group is your drug. So this protein is already associated with certain drug which we have downloaded from the PDB. Okay, but we don't want this ligand. We have our own ligand which we have downloaded from our side. Okay, we want that ligand here. We don't want this ligand here. Therefore, we have to remove this ligand. Please understand this thing. You have to remove the ligand, unselect this ligand group. You have to also unselect the water molecule. As you can see, if I unselect the water molecule, all the red droplet has been disappear. Okay, and this is now like our protein is kind of ready. Okay, once you did all this thing, what you have to do, just go on file option, save as. Now you have to save your file into PDB file. Okay, so here can you see it is in Discovery Studio files. I have to change this thing and come to the PDB file. Just scroll downwards, you will see the option of protein. Yeah, here it is, protein data bank file, which is PDB files. You have to do this change. Many of you haven't do this thing, so please see this thing. Okay, because I have already a same file with that one, I can mention one more letter double A. Okay, and don't try to mention any numeric numbers here. Even today, I guess Komal or somewhere, many people have seen they are just renaming their file name with some alphabet numbers like 55QC underscore 7 underscore 1 underscore 2. Don't do this thing. You will get error while performing the docking. So please try to take it seriously. You have to only mention the alphabets here. Just mention the protein ID underscore and alphabets. And many people what they did, they just provide space here. If you are giving some space here in naming, some most of the time Autodoc doesn't read this kind of file, so you have to mention underscore as a space if you want to give. Okay, now you have to save this file. So now this file has been saved. And if you want to do some more analysis here, like finding the binding pockets, receptor cavities, that I have also covered in previous classes. I have the recordings. I will share you by today. Just remind me if I haven't shared you with you. So you can go through it. But let me tell you here also I can do it. First of all, you have to define the receptor. So once you define the receptor, you have to go for the ligand interaction. So actually, this is the site where your drug bind with your protein. It means these are certain active regions of your protein. This is basically a drug molecule that shows how you where your protein has been interacted with your drug. So this information you have to save it. If you want to go for technical analysis, that we will not try to cover in this simple docking, that we will cover in next kind of docking. Like once you all did this basic kind of docking, for the next time we are going in depth analysis of docking, where we will cover some more aspect of docking so that we will get a precise and accurate score of the docking. So therefore, I won't want to confuse you as of now. Just go with simple one, which I told you. Just open your file, convert into PDB file by deleting the water molecules and ligand groups and save into PDB and all that. Okay. Now your work for Discovery Studio has been done. Just close this thing. So once you ready with your file, now what you have to do, again, let me show you what are the files that you have been ready. In Windows 3, I have make a folder Bioticon. Inside Bioticon, I have made two folder ligands and protein. In protein, I have my recent file which is 55qc underscore AA, and second file which is 55qc underscore. This is the file which I downloaded from the PDB, and this is the file which I have did the changes from Discovery Studio. Now this file is my final file, and these two file is of no required as of now. Okay, so I can delete these two file because I don't need this file. But you can save it somewhere else, because if you're going some other analysis for your protein, you need a PDB file, not PDBQT file. So just make one other folder here, like a bin folder. You can place these unwanted files there. Okay, later you may require. Now come to the ligand folder. Here you have to place your protein. Sorry, here you have to place your drug molecule, which is in SDF file. So all this file as mentioned below, that key your Autodoc Vina can't read the SDF or PDB file. It only read the PDBQT file. So this is the PDBQT version of this SDF file. So I have converted this thing using the OpenBabel. So I hope you've understood. So if you have any doubt, let me know. Any doubt so far?
No, sir.
Okay. Okay. Now one more thing that you have to do is active site prediction of your protein, and how you that is cast, that is cast, that is cast, that is cast field. So from this web server, we can predict what are the what are the active sites of your protein. So just simply upload your protein file here, and within a minute or 2 minutes, you will get the results of cast So you will use this cast informations in Autodoc Vina for drawing the grid box if you're performing using the Perl script. But if you're going in depth and you require a mention each, you have to mention each and every active sites there for technical analysis. If you're going to in-depth analysis, so for that you must know how we can do all this thing. Okay, so I am covering in one thing, like either you go for simple on what you have to do, either you can go for the depth analysis what you have to do. So you must require the cast analysis. So here you have to upload your file. How you can upload? Just simply click on the calculation option. Just click on calculation. So after click on calculation, you have to click on upload file option. Like here is an option. Just click on this file option and upload the PDB file. So either you can upload the file which you have downloaded from the PDB, either you can upload the file which you have did the changes from the Discovery Studio. Both files are readable here. Okay. So I have placed all my files in Windows C drive inside the Bioticon folder. Now once you upload your file, click on submit option. So actually the problem is this web server, most of the time it won't works. So you have to find out certain, substrate, or you can say as a substitute of this. Like one new web server has been found by the IIT Delhi that you can also use for the active site prediction. Okay, that I will also let you know soon, very soon. So once you get submit your protein, you will get a result. Okay, and how your result will look like it will show the result at the bottom here, and also there is one option, download option, like this. You have to click on this link. Now you have to wait until your active site is being predicted. So now the result has been loading. You have to wait. Yeah, this is the. Can you see the this bubbles, the red bubbles? So over your protein, this is the active region of your protein. Rest other part is inactive. It means rest other part is not going to, any drug can't bind there. Only the red bubble region can be employed for the binding of any drug. So we need this information of this red bubble. And what are these information? These information are this thing: POC ID, chain sequence ID, amino acids, and atoms. So these are the information. So these information you have to use in MGL tools like the Autodoc Vina, and then perform the docking. So how I can download this information? Just simply click on download. Once you download it, couple of files here, and inside that files you have to select only a certain file. I will let you know which file you have to select. Just wait a minute. Can you see right now the CASP result has been downloaded? Just wait for a minute. Just open this folder. So inside this folder you will see different files like this: POC file, POC info file, PDB file, and all these file. You required only one file, which is POC file. Again, I am repeating this thing. You required only one file, which is POC file, because this containing the pocket information or cavity information. Okay. So just opening this pocket information file. Yeah, you have to open in Notepad. Okay. So can you see the information is displayed here? Okay. Now copy all this information. Control plus A, I copied all this information and paste in Excel sheet. Here you can see there's multiple columns and all that, but we don't require all this information. We require only two columns. One is the residues and then number. Rest everything is useless. We don't require. So just clicking on blank workbook. Just paste this information here. Okay, I have just pasted here the information and selecting the column A. Now go. Just click on text. Just click on text. Just click on text to column. Okay. Now next, finish. So you can see your data is now distributed in different columns. Okay. Now I'm just selecting what the information that you required. I required this column and this column. Rest every column is useless. Again, understood this thing. I required only this residues column and then number column. Rest everything is of no use. So you don't require all this information. Just delete all these informations column by column. So I'm just selecting this thing and deleting it. Okay, so now this is the two columns which is being left. Yeah. Now you have to remove the colors which you have used earlier. Just fill, no fill. Same goes for this one. Okay. Now can you if you can notice here multiple residues are being repeated like arginine, arginine, arginine. So we don't want the repetition. We have to remove the duplicates. So how we can do? Just simply select column A, go to data and click on remove duplicates. Select the column A, go to on data ribbon. Now click on remove duplicates. So just click on expand the selection. Automatically it will click on here expand the selection. Now click remove duplicates. So you have to select the column, unselect the column B, you have to select the column A. Like based on column A, column B data will also get deleted. Okay. Now click on OK. Can you see 1277 duplicate has been removed? So after removing all these duplicates, you got unique values. And how much unique values you are getting? Only 20 unique values. So these are the 20 active sites that has been predicted using the CASP, and you will use this information in Autodoc Vina for docking part. Now you have to save this information. Yeah, either you can save here, either you can just do copy this information and paste in the biotech folder. Okay, so I'm just randomly typing anything. Okay, either you can yeah, just do one more thing, just copy this information. Okay, now close this Excel sheet. We don't require Excel sheet. Yeah, close this file as well. Come to Windows C inside the biotech folder. Yeah, here you have to make a like a txt file or notepad file. Okay, txt file, and just type here castp. The name of this file should be castp. Yes, so I'm replacing the earlier files. Sorry. So this is the blank notepad file. I have to just paste the information that I have copied from the Excel sheet. Just save this information. Now I will use this information in Autodoc Vina for the grid box drawing and all that. Now, once you did all this step, what you have to do? You have to open Autodoc Vina. I mean, and just search on this after installing the MGL tools. Just execute that file. So you have to search now Autodoc. So this is the Autodoc tools which already I have opened here. So this is the interface of Autodoc tools. Where on the initial like on the topmost you can see like 3D graphics, edit, select, display. These are the options that we required for all this docking performance and all that. So just click on the file option. Open your PDB file of your protein that you have prepared from the Discovery Studio. Just open your file here. So we have saved our file on Windows C drive inside the biotech folder, then protein, and then double A wala file. Yeah, this one 55QC underscore double A double A. Yeah, just open this file here. So this black screen shows your protein. Okay, so this is black screen shows your protein. So I will just maximize this screen. I hope it is visible to you all. So still can you see some water droplets is there, these are the structure of your protein. Now you have to edit this protein, or you can say as a prepare the protein structure. The very first step that you required for docking is preparation of the structure. So now how we can prepare our protein structure? For that, just click on edit option. The first thing that you have to do, delete the water molecules. So what the error that you are doing, most of the people, like I see what the error that I am getting from you guys, that you guys are adding charges first, then deleting the water molecules, then adding the hydrogens. So don't do it like this. First of all, you have to delete the water molecules, then add the charges and hydrogens and all that. Okay, the reason is that if I would like to show you an example with that. For example, you have to clean your house for Diwali purpose. So first of all, you have to remove the waste from your house. Then you have to, incorporate the new things in your house. If you incorporate the new thing in your house first, Then removing the waste, that doesn't make any sense, and also it will make your task hectic. Okay, so first of all delete the waste, then add the good things, or add the the things that require for the docking part. Therefore, first of all we have to delete the water molecules. So as you can see, the red droplets has been removed here. Now what you have to do, again click on edit option, go on charges, add column and charges. So this is the amount of charges that has been added here over your protein. Now next step, what you have to do, again click on edit option, go on hydrogen, add polar hydrogens. You have to select here polar only. Okay. So once you did, you can see certain droplets has been mentioned here. And, the structure of protein has also been slightly changed. It shows a blue color sprinkles like that. After doing all this thing, what you have to do, you have to open your, you can save directly your protein from here. Okay. Just click on file, save. Or you can do one more thing, just click on grid, macromolecule, choose, select this molecule, and then select molecule. Yeah, just click here, okay. So automatically you will get it open, a tab will open that shows that you have want to save your file. Just go to Windows C drive inside the Biotecon folder, then protein folder, and now this is the pdbqt file, that is our final file for docking part. Okay, now we don't have to do any other changes in this file. We will take this file for the docking part. Just every file is of no requirement. Okay, now save this file. So now our file is getting saved, and once it's saved, the color of your protein will get changed. Now what you have to do, the next step, you have to open your protein. Open protein means you have to click on this option. It will show you what are the present like chains that has been present in your protein. Now again click on chain A. You will see what are the residues that is present in your chain. Okay, sorry, in your protein. So these are the residues that is present in your protein. Now all these residues are not at all active. We have to select the active residues, and which we are predicted using the cast-p. Just open the cast-p result like this side, and just see the first active residues that we have predicted is methionine 273. Just select here methionine 273 in this rectangle box. Okay, so it will gives you some yellow color spots on your protein. Okay, same you have to go for else everything, like whatever the active residues that you have predicted. Next is 274, 275, 276, 278, like that. It means from 273 to 278 I have to that just left this 277. Rest everyone I have to select here. Okay, now come to the next one. In 279, 287. I will select here 279 and 287. And you can also, meanwhile, you can also see the changes in protein. You can see certain yellow spot has been, shown on your protein. So these are the active region of your protein that you are now using finding this Autodoc tools. After that, 287, 291, and 298. 291 and 298. And then 299 and 3013. 301 and 303. Okay, so I'm not going to do all this. Like, we have identified what are the active regions of a protein. If you can see directly, you can analyze कि only this part of your protein is active. This other part is not at all active. Okay, so try to make the grid box over this part only. Okay, and don't try to cover whole your protein. Again, I also see that most of you directly jump to 110. Let me show you what is 110. Now once you did all this active site selection, what you have to do, just click on grid, then click on grid box. Can you see a box has been formed here? So now you have to drag this box over this active region, and you have to also enhance the size of this box. So how you can enhance? Just scrolling this parameters. Okay. So for laptop, you have to keep it maximum 110. If you are running system, you have to you can go for the maximum, which is 128, I guess. Yes. So right now we are running in laptop, so we will not take it more than 110. Okay, it must be less than 110. Okay, now I will, as you can see, this yellow spot is outside of this box, but we want every yellow spot to come inside this box. Okay, we don't have to cover unnecessary part of our protein. That will only give you some false result. Okay, we want specific interactions. So therefore we have to drag this thing. So these three, the bottom offset one, these three are the scale you can see, which is used for the moving your grid box over your protein in X, Y, and Z dimension. So you can check it by moving upward here, there, and all that. If you can see, I have selected certain parts, but still this yellow spot has been left. So I will cover some more part here. Yeah. So now you can see every yellow spot is coming inside this box. So now my grid box file is ready. Once you did all this thing, what you have to do, just click on file option, click on output grid dimension file. Okay. Now output grid dimension file, that means you will get an output file which is in txt form, your grid file. You have to again save this file into biotech1 folder inside the biotech1 folder. Okay. So biotech1 folder. Yes. Here I will mention here grid. Save. So I have saved my file. Okay. Now we don't require this MGL tools and all this Autodoc Bina. Just close this thing. Okay. Everything has been done. Let me show you what file that we have generated using this grid box and all that. So this is the file that we have generated, this grid file. Just open this file and you can see the information of this grid file. Okay, like that shows the dimension x, y, and z dimension values and all that. Okay, so this information that we require for docking part. Okay, you will understand later once we are going to perform the docking, then you understood what is these informations and all that. Okay, so if anyone have any doubt in any steps, so please
Sir,
let me
I
know, so
have
that
one
from
doubt.
Monday we will move
Uh,
forward and we will try to complete
During
the first docking.
in my case, the chain was very large. Like after the data clearance and sorting in Excel, um, the resulting number of chains were residues around were 400 something. So in if in some proteins huge number of residues are coming, then what should we do?
Okay,
Should we like yeah.
okay. First of all, let me tell you. Yeah, yeah. So let me tell you what the things that you are might doing wrong. First of all, you have selected all the chains of your protein. So you can take it one chain at a time. For example, if my protein is consisting of five chain, like chain A, chain B, chain C, D, E, and F, like that. So for the first time, you can take chain A and just upload that chain A file into Caspi, and now you can see the residues has been, the number of residues has been reduced. This thing you can do it, okay? And same goes for all these other four type of chain that you have taken. And once you have the score of all these four chain or five chain that you have taken, you can see which chain has given you the maximum or
Yeah, yeah.
maximum negative score. It means the better binding energy score. You can
Okay,
select
sir.
only
I will
those chain,
try
and
and
left everything you can discard it.
I will try
I
and
hope you
then
get it.
let you know if
If you not, then you
yes.
can share your screen.
Thank you,
I can
sir.
let you know. If you have still any doubt, you can share your screen.
Sure, sir. Yeah.
That's also. Yeah, yeah, sure. Please text. No, also you can text me anytime if you have any queries. I will try to answer each and every queries of yours. Anyone else have any doubt?
Hello. Yes, I understood.
Okay, hi Jonty, could you please unmute yourself? Did you understood? Hi, did you understand?
Okay.
If you have any doubt, please
So
let me know. I
I'll
can
share my
yes or
screen.
either you, if the good if you can also perform by sharing your screen, that could also be the great.
So from first, we choose like we choose a protein from this PDB, this RRCSB, correct? And then we also choose like a ligand file, like it could be a drug or something. So ligand can be chosen from PubChem, am I right? Okay. Okay.
Yes, but let me
Okay.
correct you. You have to choose the ligand. Ligand is also called as
Okay.
your phytocompounds or your compounds like that. Okay, you have to use certain terms. Like your protein can also be called as receptor. If you can go for a stimulation one, you can see their receptor ligand
And
interaction or protein ligand interaction. So don't get confused. Your
phytocompound.
protein is also called as receptor, and your ligand is also called as a compound. Okay, or phytocompound. If it is from
Okay,
a
drug
plant, you can
candidate.
say as a phytocompound. And one more term for your ligand. As of now, I'm not
Okay.
remembering what is that term.
So
Your
I
drug. You
don't
can
know
also say
which
as a drug candidate like that.
like
Okay. There are
currently
like
which
many and many terms you can
protein to
say.
select
Yeah.
which you had.
Please continue.
I was trying to find out like can you help me out here, like which protein to select? Okay, my screen is not, sorry. Uh, just a second.
Yeah, you can share your screen you can share your screen. I can guide you here. Okay.
Is it visible now? Okay. So here I am in this correct PDB protein bank.
Yes,
Okay. I I don't know any protein. So that is
you have to type any protein that you want to take for docking. So any
o-
protein, if you know, you can take it.
okay.
Okay, so we have taken from our side
Okay, this one.
RNA dependent RNA polymer. This is a viral protein which found in
Okay.
dengue virus, and
So
for
we
this we require any antiviral drug.
no, I don't know.
Yes, you can take. Yeah, yeah, correct. Did you know why it is
Hi-
mentioned hydrolase and all that?
Yes. Yes. Yes.
Okay, did you heard the enzyme
Okay.
classes?
Okay.
Transferases, hydrolases, ligases, each seven classes of. Okay, try to study this thing, then you can
Yes.
understood.
Yes.
Okay, so directly you can't take
Upward.
it here. Just go to bottom and
Okay.
explore the protein structure.
More up.
Could you just come to the bottom? Five
Okay.
clicking. Yeah, can you see here? No, no, no. Go upward. Go upward. Yeah, little more upward.
Yeah. Yeah. Yeah.
Yes. Still, you
Yeah.
have to
Yeah.
go
Yeah.
some more upward, where you can see some, angstrom values is there. Yeah, yeah. Can you see here the method is mentioned here, experimental data snapshot.
Okay. Okay. Okay.
So here you can see the first thing that is method. It means your protein has been found here using the X-ray diffraction method. If you have guys know that was a different tools or
Okay.
experimental method that required for your protein structure
Prediction.
founding like crystallization method and many more methods. The best
Okay.
one is this one, X-ray diffraction method. Okay, that means your protein is the best prediction. Okay. Second thing, the resolution must be less than 5 angstrom. If any protein having a resolution is more than 5 angstrom,
Okay.
you can't take for docking. It will
Okay.
gives you a false result or it undergo certain problems for the
Five.
publication.
I understand.
You have to always go for resolution less than 5 angstrom. Okay. Remember,
Okay.
resolution must be less than 5
So
angstrom. If these two parameter has been followed,
dengue virus. Yes.
yes. If these two parameter has
Okay.
been followed, directly download your protein into PDB file. And can you see it
Okay.
is in dengue
Okay.
virus?
Thank you.
Okay, that
And
means it's a
now I
viral
need to
protein.
download my
It's also
uh
pathogenic protein that
okay.
causes
So
the dengue
it
disease
was
or dengue fever.
I need to download my drug. So it could be so uh hydrochloroquine. Uh
Yes, good. No, no. You require
okay.
any
Okay. Okay.
antiviral drug. If you know any antiviral drug, it means any drug which is being,
Yeah. Yeah. Yeah.
binding
Okay.
with any viral proteins or viral genes like that. So remdesivir is an antiviral drug, cytobudine is an antiviral drug, lamivudine is an antiviral drug. Okay,
Okay. Okay. Remdesivir.
so you can take zidovudine.
This
We
one.
have taken zido. Either you can
Okay.
take remdesivir. Yeah, you can take it. Okay, we can
Okay.
also
Okay.
see different score. You can go for remdesivir. Try
Molecular.
for it.
Yeah.
Yes, correct. Yes.
Yes.
Wait. Again, wait. Can you see here molecular weight,
No, I've not heard about it.
which is 531.2 g to 2 gram per mole. So,
Okay.
have you heard Lipinski rule of pipe and Weaver's law?
Okay.
Okay, just read about these two
Okay.
kind of law. So actually, if any drug having a molecular weight of more than 500 gram per mole, you can't take that drug as a
Okay.
oral. Okay, you have to
Okay.
go for the penetral route. So always try to take the drug which is less than 500 gram per mole if you want to go for oral bioavailability.
Okay.
I hope you got it. And if you are going to perform using the injection mode or penetral route, you can take it like certain greater than 500 gram per mole. Okay.
The
But right now we are learning the
this
docking, so you
download
can take
button.
anything. There's no any objection. Just download this. Just click on the first one,
Oh,
remdesivir triphosphate one,
okay. This file. Yeah.
the blue one. Like yes,
Okay.
blue. No, no, not here, not here. Open the this file. Yeah. Just open that.
Yes. Yes. Okay. Here. This
See
one.
some more information is here. Could you just come downward where you can
Yes.
see
Yes. Okay.
canonical smiles? This is the structure of your drug.
Okay.
Then IUPAC, yes. Then
Yeah.
IUPAC name.
Yeah. Yeah.
Then
I've heard.
income. This is the income
I've heard.
formula.
Mm,
Okay. This is the income key, in Campbell keys.
okay.
Have you heard Campbell database? Campbell database like that.
Commendable.
Okay. So these informations has been predicted from Campbell data. Okay?
Okay.
Just come to yeah. This is information important. Canonical smile. Okay. So once we perform the docking, we have to go for certain other analysis. For that, we require the canonical smile. So this is the you can say the alphabetical informations of your drug.
Okay.
You will use this canonical smiles and go for protox, for toxicity predictions, and oral viability prediction, and many things we will do using this canonical smile.
Okay. Okay.
As of now, we don't require this canonical smile, but keep in mind that this canonical smile is very, very important for
Yes.
all
Yes.
kind of analysis that we
Yes.
will
Yes.
do after the docking. Okay. Now, can you see on the
Okay.
right hand
SDS.
side at the topmost download option
Okay. Okay.
after the site option? Just click there and download in STF form.
Yeah. Okay.
Okay, one more thing. Can you? No. Wait. See here, like your drug consists of only
Okay.
2D structure. It is not consist of 3D structure.
Okay.
Okay.
Fine. Understood.
So, but you can't take it for docking. For docking, we require the 3D structure. Therefore, you have to change your drug. Understood? You for docking, ideally we require the 3D structure. If you take 2D structure, you might get a
Okay.
problem in structure conversion or something like that. Therefore, always I'm mentioning you have to take 3D structure. Again, take any other antiviral drug and download the STF file just like that. Okay. So you can go for any other. You can take lamivudine. It's also an antiviral drug.
Yes. Yes. Yes.
Yes.
Okay.
Can you see the molecular weight is being here 229 gram per mole? It's less. And you can also see the
The
yeah. So
structure.
you can take it as a oral viability test like that. Just open
Okay. Yeah. 3D.
this one. Yes.
Okay.
Okay, and now again go to downloads option.
Okay. So now I need to make a
Can you see the
folder.
3D structures? Yes,
Yes.
you can download now SDF file.
Um,
Correct. In Windows C.
Here one I'll keep my drug and another my protein. So I'll shift my protein uh, to here, and my drug should be okay. So I have placed my drug and my and my protein. Now I need to uh, mm, now I think we need to uh, check it. So kernel structure discovery studio. Yes, yes, here it is.
Yeah,
Discovery
we
Studio.
have to explore the internal structures of this protein using the Discovery. Yes, correct. I hope you have
Okay.
downloaded the Discovery Studio. Just open Discovery Studio and open your file. You can also drag your files.
Oh yeah, I've installed Open Babel and yeah.
Okay, have you installed the MGL tools
Okay.
and Open Babel?
Okay. Okay.
To show you how to use the Open Babel, but
Okay. So I can just drag and drop. Okay. 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 So
yeah, you can drag and drop, either you can open your file. No, just correct. Hmm. Now just click on the option, this menu.
yeah, yeah, yeah.
No, no,
And
not
yeah,
there, not there. Just ahead
yeah,
of
yeah,
the
yeah, yeah,
analyzed
yeah, yeah, yeah,
transmembrane
yeah,
protein.
yeah,
Can you
yeah.
see a small arrow is there? Analyzed
Okay.
transmembrane protein. No, no, not there. The
Yeah.
bottom, the last one. Analyzed transmembrane. Ahead of, yes. Yeah, exactly. Just click on
Black
this
screen.
option. Now you can
Screen
see
protein.
like your protein is consisting of chain A.
Okay.
And could you just rotate your protein? Just click on the
Yes,
black
yes, yes.
screen and rotate your protein. It is not a, just double
Okay.
click, double, yeah, you can able.
Okay.
Can you see there is a ligand here inside this protein?
Okay.
You can see.
Unselect.
So that we don't want, because your protein is already occupied with your drug. We have to remove this drug, okay, from here. So just
Okay.
unselect the ligand
Okay.
group.
Okay.
Yes, now your drug is, just
File.
unselect the water molecule as well, because
Save.
this water molecule causing
And save
hindrance
as. Okay.
for binding, okay. Now you can save your protein, okay. Just again click on file option,
And
save as,
PDB,
save as,
and just put a single
and now change the file type. You
underscore
want
a.
in Discovery Studio. Yeah,
Underscore.
go in protein folder, very good.
Okay. Okay.
Now,
Okay. And then save it.
yes, correct. And just underscore a, yes, to differentiate from that, okay. We are doing this thing,
Okay.
yes.
Okay. So
And there is some more
now
in
go,
studies there that I will show you
now
later,
I
or I
go
will
to the
show you,
server.
share with you some recordings of previous classes. You can follow
Uh,
that one as well, okay.
the server
Now
was called.
your protein is a, yes,
Oh, sorry. Cast P. Okay, thank you. Cast P. Go
Cast
to
P. Just remember
calculations
the word cast P.
and then upload the file. This one.
Calculation.
Okay.
Yes.
And this is all 1.4.
Hmm.
And okay, submit.
Yes.
Okay. Okay.
Yeah, you don't have to change it. Submit. You have to click my 2 to 3 times because it's not fun. Yeah, it's work. Just click on this link.
Okay. Okay.
So your protein is quite
Yes. Yes.
small one, so you get a result
Active.
within a second. If your protein is
Okay.
complex, it might take 10 to 15 minutes, okay? Because this protein is simple, so it can easily predict it. Can you see the red bubbles inside your
Okay.
protein? These are the active parts, okay?
And
Where your drug can bind.
POC file.
Now go upward and download the data of Caspi.
POC file and open it in Notepad. Okay. Always. Now I
Okay,
copy
this
this
one.
in Excel.
Very good. BOC file.
And go to data, uh, text to column, next, next, and finish. And then I go to remove duplicates. Uh, so I go to unnecessary columns here. So column D and column F.
First
Okay,
of
this
all,
one.
you have to delete the unnecessary columns. Okay, like column. Yes, you require only column
Okay,
D and
okay,
column F.
okay,
No, no, no. Like it's right now changing. You can change the color. Okay.
okay.
Yeah, that's also good.
Column A, and
Yeah, yeah. Now it's correct.
then
You can
I
delete all these
go
selected one.
to
Now this is the information. Now you have to remove the duplicates. Select the column
do I
A.
go here? Do I go to data? Okay, where do I go?
Hmm.
Okay, so I forgot how to do. Mm,
No, not filter. You have to delete
okay,
the
go to data.
duplicates. I think there is no.
Oh yes, yes,
In
yes.
select the column A, go to data.
Expand the selection. Just select
Here you can see
column
somewhere
A
remove duplicates option.
and then press OK. Okay,
Yeah, this one.
1277 duplicates values found and removed. Okay,
Yes, correct.
okay, okay. Then I okay,
So these are the unique values that has been left here.
and go to
Now you have to save this information and paste into
and
notepad
make
file.
a new called cast P.
Copy the information of the, yes, copy all this.
Okay, and you can save this file. Okay, now open Autodoc
Save this
tools.
file, save this information. Okay. Now you, yes, correct.
Mm, now I can open it from here. This PC, this one. So this is my structure. Firstly, I need to remove the water molecules, and then add Coleman charges, correct? And then I add hydrogens, polar hydrogens. Polar, okay,
Correct.
Okay. And now I need choose, right? Like that.
Polar hydro. Yes, correct.
Okay.
You have to save
And
this
I
file.
save. Okay, it changed color. And now I need to select the uh, look at this and then select the molecules, the active sites. Sorry, active sites. Yeah. And then after selecting,
Yes, not
okay,
molecules, active site, correct.
73, 74,
Just drag your
75,
notepad. Yes,
76,
correct.
76, 78, 79, 87, 81, 98, 99. 307, 311, 326, 339, 349, 354, 387, 446. Okay, selected all. And then again I go to grid box, and then I select only the active sites. I make this bigger. Okay, I can't see anything. Mm, it's not selecting everything. Okay, I'm running laptop. Okay, I'll.
You can, I told you, na, maximum you can go 110 for laptop. If you're running in laptop, you can go maximum 110, okay?
Okay, okay, oh, okay,
Correct. Now your
okay.
grid
Sorry.
box is finally ready.
On
Now
this
you can
grid
save
table
this
and
file, okay? Grid dimension
okay,
file.
okay.
Just click on the file and save it.
File
No, not that file. On this grid table.
third output.
No, don't
Okay,
close it.
output. Okay, okay, okay.
Yeah.
Okay,
No,
and
no, output grid dimension file.
in the
Output, the
proteins
third
section
option.
only.
Output, again click on file. Go to Windows C where you have saved
Okay,
biotech, where you have created biotech
okay.
on.
Grid file.
No, no, not protein folder. You have to go in Bioticon folder. This is independent
Okay,
of your protein and drug. Here you have, yes, correct.
okay, okay, okay,
Yes.
okay. It's created.
So now you can close this Autodoc.
Oh,
Then first of all check whether it is being created or not like
one
that.
thing.
Then you can close this thing. Now you are
Okay,
ready with your protein, your
okay,
active
okay,
sites, your grid file. You are
okay.
left
Thank
with
you.
one thing. your drug should convert it into PDB-QT file. Okay, it's just a 1 minute or you can 30-second work that I will show you on Monday.
Okay.
Okay.
Thank you. Thank you.
Thank
Hello.
you, sir.
Hello, sir. Sir, what about the CRISPR
That's all
you
for
sent?
today.
No.
Now
What
your
about the
call will
timing of
begin.
that?
So thank you for joining. Hi. Yeah, actually we plan in evening
Because
time,
both
like
if you taking no
like
one on the
between
other
6
way,
to
it
10
will
we are
take time.
planning.
No,
Like
that's what
most of the people have
I'm
saying
thinking
that
in
to
that way
plan in
also.
evening from 6
This
to
class
10.
also is
So
running
like
in
that.
one time, and that might be clash also there, or we can't perform both.
Yeah, yeah. No, no,
Oh,
no. Yes, like docking class will not be there from
okay, only
20
3 days is there.
on that day, 23, 24, and
Okay.
25. We will try to complete it before, like I will take some extra time as well. So on docking class will not be there on that time. Only Christmas session will be.
Okay.
Yeah,
Okay.
only three
Fine.
days will be
Fine.
the
Fine.
Christmas session, and it will be
That's
like
the only
full
clarify.
hands-on. Hands-on means every
No,
each
no.
and everything we'll
Because
try
of
to
that only,
explore
I'm
in
thinking
of
if it
usual
is
whatever
clash,
has
then
been
I
done
how can I
computationally.
perform both in that way? So I'm thinking whether I have to register or
So
not.
have
Like
you registered for the
now you are
workshop?
clarifying. No, I will register for that. Okay. Okay. Thank you.
Yeah, yeah, got it, got it.